Okay, great. Okay, I guess we'll get started now, everyone. So this is Dr. Grace Mead, who is actually a postdoc working in our lab, the Planetary Exploration Lab in Aerospace Engineering. And she got her Bachelor of Arts in Geoscience at the Franklin and Marshall College in Lancaster, Pennsylvania, and then got her doctoral degree in astrobiology and geochemistry, or mainly geochemistry, at the University of Maryland in College Park, Maryland. So she did a lot of really cool research in the detection and characterization of biosignatures with laser-enabled Orbitrap mass spectrometry, which is really exciting. I think you're going to tell us a bit more about that today. And then in our lab currently, she's using bubbles to concentrate things, which is really cool work as well. So I think you might touch on that a little bit today. So here's Grace.
All right, thank you. Thank you, Rachel, for introducing me. Now that just makes me extra nervous. But it's okay, we're gonna get started. And today is International Women's Day for those on the Zoom meeting. So hooray, if you are a girl, and then we're gonna celebrate for each other. Yes, and then like Rachel said, I'm a postdoc right now. I work with Chris Carr from the AE and the EAS department, as well as Facundo Fernandez from the chemistry department. I just started my postdoc position, so today's presentation, I'm gonna primarily focus on my PhD work, and I will introduce a few slides about my ongoing postdoc work as well. So like I said, my PhD work focus on characterizing chemical signature of life via mass spectrometry. So you want to search for life elsewhere within our solar system. You can either go with a lander or orbiter, but no matter which way you're going to go, you probably want to design an instrument. Well, you want this instrument to be as small as Ant-Man, as ruggedized as the Hulk, as reliable as Captain America, as versatile as Iron Man, and as best performance as Captain Marvel, right? And when all of them combine, hopefully, you can design ultimately one instrument, that's ready to fly high to the sky, ready to be launched to outer space. Now, that process, to start with, it's just like how you play Pokemon Go. You start with an idea, and with many years of development and testing, eventually that will reach the ultimate spaceflight instrumentation goal. The thing is, there are a lot of things that you can play with. During my PhD, I've been encountered many instrument prototype, and then that just span across different regimes. Among these stacks of cards that you can potentially play with, the one that I'm focusing on are mass spectrometry. And then you may wonder, what is mass spectrometry? So mass spectrometry is a technique to identify chemicals based on mass to charge ratio. So how you think about it? Don't think it too complicated. Although the field has been expanded for a broad application, but in general, the fundamental rule is that if you have a compound, just like histamine here, and it's composed of 6 carbon, 9 hydrogen, blah, blah, blah. And then if you can add the mass of individual atoms together, you will get a very precise value of the mass that this molecule is represented. Mass spectrometry, or a mass spectrometer, which is the instrument that uses mass spectrometry, is able to measure those precise mass, and using the mass to infer what is there, what are the chemical constituents of the unknown compound. So that's the fundamental rule. Mass spectrometry has been used for space applications since the early 1970s, and then has been still being using today. There are rich history of mass spectrometry in the planetary exploration. In general, there are 3 primary components of a mass spectrometer. You have your sample that comes with neutral, right? There's no charges. And then you will first go through an ionization source. Basically, it's just a simple subsystem where you can put charges on your neutral analyte. This analyte can be gas, liquid or solid. After that, you have your charged analyte. You want to separate them. You want to manipulate the ions in a mass analyzer. So you want to separate a mixture of ions based on their mass to charge ratio. Once you do that, you want to measure or quantify the amount of ions within each specific mass to charge ratio. And the output mass spectrum basically give you the intensity as a function of mass to charge of the analyte, right? There's nothing special about this technique. So within each subsystem, within each ionization, mass analyzer, detector, there are multiple technology going into it that give you different flavors of mass spectrometry. In reality, when we are dealing with unknown sample, they can often be very complex. So in addition to ionization, mass analyzer, and a detector, people often hook up with a hyphenated technology. Things like, I want to convert solid into gas. I want to convert a liquid into gas. Maybe I need to employ a pyrolysis oven or laser sampling. There are also separation techniques like chromatography to separate a complex mixture into different distinct group of analytes. And then you inject them into the mass spectrometry. But here we group all of them together and call them hyphenated technology. There's nothing special about how you design a mass spectrometer. You can basically select and mix and match to build a different mass spectrometer. And let's do that. Just like how we create our own pizza, you select your own toppings, okay? So back in the 1990s, people trying to emphasize the composition of atmosphere, and they target gas mixture. They use a gas chromatography to separate gas mixture, inject it into electron ionization, and then analyzing them via quadrupole, and then detect them via electron multiplier. Those are just the term. You don't have to pay attention or remember those if you're not in this field. But basically, those specific selection of technique comprise an instrument called GCMS. And in the commercial realm, this has been developed by Thermo Fisher, and it's large, usually cover the entire bench, and it can be 78 kilogram in terms of weight. For space application, we try to miniaturize it to a much smaller size. So these are 45 times smaller, 4.5 times lighter, and then use an average power assumption, 41 watts. What does it do? This mass spectrometer basically has been put in a probe that's been in the Huygens probe and be used to understand the atmospheric composition of Titan, which help us to collect very preliminary understanding or analysis of Titan's atmosphere and solid particles, which ultimately motivate a new mission, Dragonfly, to visit there. Later on, when we're trying to target Mars, for example, now our target analyte, instead of gas, we are looking forward to analyze or get chemical composition of solid and primarily sub-Martian core or surface soil sample. For those, we will need a conversion from the solid particle to the gas molecule. That's when we add an additional pyrolysis oven to the instrument. So this is pyrolysis GCMS, again, commercial instrument. And this is what we propose for SAM's instrument that's currently on Curiosity rover. As the time progresses, now in the recent time, we are trying to develop something more advanced. This is because the evolution of the technology, as well as the scientific questions, become more diverse. Now, instead of just looking at a single type of compounds, we want to explore even compounds in different phases. We want to add more versatility, into the instrument so that we are able to tackle compounds in different form, in different phases that collected in different settings. And because of that, now instead of just focusing on gas phase molecule, we are also focusing on solid phase and using a laser to sample the solid at a high spatial resolution. So this is one ongoing instrument actually gonna deploy on the ExoMars rover. So this one, you basically combine two different ionization sources to tackle different type of molecule and all adding up into the instrument. So the primary takeaway of this session is it really depends on your needs and then the targeted sample. You can build spaceflight instruments based on how you want to analyze the sample, where you're trying to deploy them, and by piecing them together, you are able to create a new instrument concept. Now, development of spaceflight instrument can often take decades. This is because we have limited resource on the spacecraft, and that often we are faced constrained by size, volume, mass, and the data storage. So that often requires miniaturization of the commercial instrument. Because of the unique space condition, vacuum, microgravity, launch environment from the shocking, we also need to ruggedize our instrument. Many of the system design that we normally do on Earth, we cannot apply them for space directly. This is because the unique set of space condition. We also need to consider planetary protection. And then because of each subsystem, they need to work on a system level. We have to do lots of iterative testing of the subsystem and then piecing them together to demonstrate the ultimate performance in a simulating chamber. So at the beginning of my PhD, I was lucky enough to work with one of the instruments called POROS. So this was back in 2018. POROS called characterization of ocean residue and life signature. So basically, it's an instrument prototype that combines laser ablation mass spectrometry with Orbitrap mass spectrometry. This instrument is about 2% volume and 4% weight compared to the commercial counterpart. So it's much smaller by its size by the time when I get it. Why laser desorption mass spectrometry? A focused laser beam allows direct access to a wide range of analytes. This includes organics, oxides, minerals. It really depends on the selection of the laser wavelengths and then the laser energy you are choosing. They have access to excite a wide range of molecules. By play with the laser intensity, you can not only just desorb the organic molecule, right? You can also tend to fragment them at a higher laser energy. A focused laser beam allows you to ablate across the sample surface, and then you can establish chemical mapping and trying to understand the distribution of a specific chemicals across a 2D image. Orbitrap mass spectrometry is one of the latest mass analyzer that's being developed to detect and characterize chemical unknown. This instrument, it requires pulse injection of ions that being produced from the laser. Once the ions being injected, they will be starting oscillating around the center electrode at a frequency that's proportional to the mass to charge ratio. So this is very important because the really fast oscillation back and forth along the center electrode allows a really high mass resolving power. In other words, if you are not very familiar with mass spectrometry, basically what this does is it allows us to measure the mass measurement, mass to charge measurement of the unknown compound back to the 4th decimal place instead of the 3rd or the 2nd decimal place. So think about it. If I have an unknown species, now I have the capability to measure to 100.0000 instead of 100. The back and forth oscillation will be detected by the outer electrode, which will be output as image current. So the top image shows you the raw transient signals. This is time domain signals. But once that being Fourier transformed, we're able to generate frequency signals. and then we calibrate them to produce mass spectrum. If you have to get one takeaway from these slides, is that Orbitrap mass spectrometry give you a really precise mass measurement that help to enhance chemical identification. So when I first got this instrument was already designed for lander application. Basically, the entire instrument will be fit on a lander and drop on the Europa surface. Sample will be directly collected from the Europa surface, dump them into a sample cup, and then delivered to the instrument. The laser beam will just shoot on the sample surface. Ions that generated from the sample surface will be injected into the Orbitrap. Here's often the dilemma that I find in my team. The scientist I want more science, but then the engineer says, okay, most of the design has been set. There's not much I can change. So as a middle person in between them, sometimes I need to figure out, okay, with the current design, what will be the clever way to operate the instrument to advance the analytical technique that helps me to address more scientific questions. And then those concerns or those questions actually drive to the development of my research projects during my PhD. One of the questions I want to ask is, can we actually detect peptides if we acquire sample from the surface of Europa? So our collaborator, Brooke and Karen, they are microbiologists from the University of Washington. So they basically went to the Arctic Sea, and they argued that microbes, if they exist in a similar environment on the Arctic Sea, which is highly saline and then sub-zero temperature environment, in those environments, they are analogous to the environment that could be habitable on the surface of Europa. If we are able to find life and then there are similar peptide sequencing in the microbes that survive in the Arctic Sea, maybe that can help us to identify the potential biosignature on the surface of Europa. So basically, they acquired sample from the icy brine that in the Arctic Sea. They culture the microbes in a different controlled environmental condition in the laboratory. And then they profile the peptide sequencing or the peptide expression of microbes that tend to survive from those environments. By doing statistical analysis, they do narrow down a specific set of peptide sequencing, short peptide, usually combines of 3 or 4 amino acids together. And they argue those short peptides could potentially be the biosignatures if any microbe survive on the Europa surface. My question would be, can a laser beam be able to detect, sequence, and characterize those peptides, right? The challenge I will face is I can only play with the laser beam. You can imagine if I have peptide being detached on the surface, if I'm operating the laser at a very low energy, those molecules will still be able to stuck on the surface and never be able to desorb or lift it from the surface. If the energy is very high, I may potentially fragment them. Then how should I proceed? Challenge 1, how to desorb, right? How to desorb those larger molecules, polypeptides, without extensive fragmentation. So in the mass spectrum, you can see here, I'm showing on the right, M represents a specific peptide or a larger molecule. Those molecules can be detected by attaching a proton or sodium or in different phases. And those peaks help me to identify the stoichiometry of the unknown compound. In the commercial realm, in order to avoid using laser to fragment those larger molecules, people tend to add UV absorbing organic matrix in order to enhance the desorption of a larger peptide. However, organic matrix are a little bit concerning. So in terms of my project, instead of using typical UV absorbing organic matrix, I try to explore silicon nanoparticle. Let's say if my potential targeted analyte is a peptide. Peptide is a sequence of amino acids that's being linked together, right? If I'm able to identify the stoichiometry of peptide, it's hard for me to know what will be the sequence of amino acids within the molecule. So in order to understand the sequence, I still need to fragment them. That leads to my second challenge, how to achieve control fragmentation of molecule in order to produce diagnostic fragments to inform the amino acid sequence within my unknown. In the commercial realm, we usually use a collision cell in order to achieve that. But here, we are not adding additional subsystem to what we have already been proposed for Coral's instrument. Instead, we just want to play with the laser energy to achieve fragmentation from the ionization source. So this is the prototype of breadboard instrument that I was playing with. So this breadboard is currently at NASA Goddard. Basically, we want to mimic the sample preparation scheme that we will eventually use when we perform the instrument in the Europa condition. So basically, we prepare a peptide solution, drop test them into a metal plate, and then trying to use the laser to directly detect them. We will wait until the solution desiccates to produce a thin residue. Eventually, the laser will ablate at the residue of the organic molecule. For some part of the study, instead of just depositing solution onto a metal plate, I also added some silicon nanoparticles and repeat the similar process. And this is how they look. So here's the spectrum. So the first spectrum, so the target analyte that I'm looking at is a former peptide, oligopeptide. It combines of 4 different amino acids, and together is called GDAE. Each character represents a specific amino acid. This analysis was performed just on the metal plate without any silicon nanoparticle. And the spectrum, mass spectrum on the left, showing you a single shot analysis. The peptide was observed by attaching with a sodium. So the ideal case is that this schematic showing you that organic was deposited on the surface with the laser beam interacting with them. They are slowly heating up and then fragment them in the air, in the plume. In order to control the amount of fragmentation or enhance the signal intensity, I added silicon nanoparticle in order to heat up the analyte much, much faster to achieve rapid heating. In this case, the rate of molecular degradation will be slower than the rate of evaporation, which means the molecule are evaporated, lifted up, desorbed from the metal surface into the laser plume, more intact, faster than the rate they're gonna degrade or fragmented by the laser interaction. By doing so, I was able to see a higher intensity or more variety of organic adducts that helps me to identify molecule. At the same time, I was able to achieve lots of characteristic fragments that allows me to sequence the peptide. The addition of nanoparticles also enhance the reproducibility of the analysis. So in a neat sample without any nanoparticles across 240 different spectrum, only 6% of the spectrum, I was able to identify the unknown compound. Whereas with addition of nanoparticle, I was able to achieve those effective scan. The scan with molecular species increased to 64%. So that's ideal. Primary takeaway from the project 1, the best way to proceed in order to detect and characterize peptide in space, maybe it's just to engineer the substrate. By embedding nanoparticles on the metal plates that we have on the sample cup, and then directly deposit IC sample on the surface of the cup, I was able to achieve more reproducible, enhanced signals for peptide detection. The second project is about molecular size. Basically, these projects is deriving from, for example, if we know the chemical compound, but sometimes two compounds, they have the same chemical composition. They give you the similar mass to charge ratio. So in a single mass spectrum, they appear as a similar peak. However, the atoms in space, they can arrange in a different shape. Just like the figure I'm showing you here, some atoms may group into a sphere, whereas some atoms, they may group into a linear chain. Although they have the similar mass, but they arrange them differently in the space, so they represent different molecules. If we just have mass spectrometry, how can we potentially dissolving them apart? One way to think about this is just like how you would like to play with a chocolate bean. Imagine we start with three different types of chocolate. We can play with them and arrange them in different shapes. Now, in a two-dimensional frame, y-axis is the total area, and then x-axis is total mass. You can imagine you can start to play those molecules on this 2D dimensional space. You start with a linear chain with milk chocolate, and then because the peanut chocolate, they are larger, so you can imagine they have a larger total mass, and then you shift to the right. And then because the area-wise, they're still linear, so maybe the area in 2D dimensional space doesn't change much. This will change significantly, however, if you start to put them in a circular ring. The idea is that different class or characteristic or chemical group of molecule, they may fall on a linear train in the CCS or in the area versus mass range. And the linear train is corresponding to the density and the structural compactness of the molecule. Now let's replace those chocolate beans into TROPS element. And again, we can arrange them into different class of molecule. This type of study have been done numerously in the chemistry. People trying to understand how different class of molecule, they can fall onto different chart. So here, the CCS, collision cross-section, is similar to the area that we were talking about. CCS means the effective area around the ions where the momentum can be transferred upon collision. So basically, it's like collisional size area of an ion, right? So what happened is during the orbit trap, when ions inside the orbit trap are oscillating, they are generating mass spectrum in the way that we were talking about. However, during their oscillation inside the orbit trap, they will eventually and actually inevitably bumping into the residual gas molecule because there will be specific, for example, nitrogen gas in the orbit trap background, and one single collision will lead to ion losses. So over time, the transient signals that we collected from the ions, they will decay. Now, different decay rate will tell us the different size of collision or cross-section area. So the ultimate goal here is trying to understand or trying to use the decay rate of the transient signal to infer the collision size area of the unknown compound. How are we going to do that? So at the beginning, we start with the transient signals, and we process it with Fourier transform to generate mass spectrum. This is the typical step that we usually do with spectrum that's being produced from orbit trap. what we do here is to create snapshots of or segments of transients, cut them shorter, and sliding them across the finite transient, right? So I will produce mass spectrum from each segment of the transient, from each slice. And then by aligning those short segments of mass spectrum over time, I will be able to rebuild the decay profile of intensity. Now, one thing I can do is to fit the decay or fit the peak intensity of this consecutive mass spectrum over time. I'm able to derive a decay constant. And then this decay constant will be correlated to the collision cross-section area of the ions. By doing so, by doing this similar procedure across any identified peaks in the mass spectrum, which are labeled red circle here, I basically can derive derive decay constant or CCS value of any peak that I'm interested in the mass space. By using two of them as internal standards, I'm able to convert the decay rate to CCS value, if that makes sense. So to implement this experiment, I use a transient signal that collected from two instruments, the Q exactive and LTQ Orbitrap. The solution or unknown sample that I'm looking at is a CalMix. So basically, the Ultramark is one of the primary components in the calibration mixture of Orbitrap instrument. This type of molecule, they are kind of like a molecular complex where the N represent different groups of additional complex molecule can be attached to each branch. This is similar to you start with a ring molecule, but then as the N change from 1 to 3, you start to expand the molecule larger and larger. In the same solution mixture, I will have two former peptides. They are different sequencing here. This will be one representative transient signals that I collected from the Orbitrap mass spectrometer. So by just performing Fourier transform, I'm able to generate mass spectrum of all identified species from my solution mixture. They are labeled by color and shape. You can see that for the Ultramark, because the N changes, because they can also fragment, we actually generate a range of ionic species across from 900 all the way to 1900. So for any peaks that being identified in the mass space, I can generate decay profile using the slicing method. And by modeling them, I'm able to derive a decay constant as well as derive a CCS value. If we are able to put them into the CCS to mass to charge space, you will realize the peak or the decay constant from the same compound, they will follow a linear line. The trend line of those, the slope of those linear lines are summarized in a box plot. So I repeat this analysis across 14 different consecutive analyses, and then we can see a separation between the linear oligopeptide versus Ultramark. Ultramark have a much gentler slope in the CCS and the mass to charge ratio. This is because the molecule or the atoms, they are arranged in a circular ring. They have much denser structurally. For any increase in mass, will have lesser change in the size or collisional area. Whereas if we are talking about linear chain molecule, like the peptides here, any increase in the mass will associate with a much larger change in the CCS value. This is how. Even for the same peptides, we do see some separation between their linear slope, and that could be argued by the change or variation in their side chain As you can see, GDAE and the MRFA, which are the two peptides that we're looking at here, they have different side chain residue being attached to the amino acid. And because of that, their collisional cross-section area are slightly different, even though they are all arranged in a linear chain. Now, this is my PhD work conclusion. To tackle or trying to figure out how to use Coral's instrument to analyze and detect peptides, maybe the takeaway is to add nanoparticles in the substrate and then use the laser to operate and analyze analyte at different energy in order to achieve simultaneously detection and the sequencing of the peptide. The other one is if we only have the transient signal coming out from the orbit trap without knowing any additional more information, to expand the existing toolbox of the orbit trap mass spectrometry, what we can do is to sliding across the transient to derive the decay constant and understand how use the decay rate to infer the structural compactness, the atomic arrangement of unknown compounds in the mass spectrum. Now, towards the end of my PhD, I would say, because remember starting in 2018, I was looking at a very initial instrument, but now with the development of new technique, we would say the instrument has been advanced or mature at a higher level. And this work, of course, is being done with a large team of people, scientists, and engineers from NASA Goddard, from UMD, as well as from our French collaborator. The eventual size of the orbit trap is actually really small, including the laser box. In total, the dimensions are here. Now, that was my PhD work. And then now, as I get into the postdoc work, working with Chris Carr, I continue to pick a different instrument car. And now I'm starting with a new instrument, which is called BOOST, bubble-based ocean world organic square sample trap. So this is a new instrument design. We are in a very early phase. We want to demonstrate the functionality of this instrument prototype. The idea is that the science motivation is if there are cell or organic molecule in the ocean world sample, they are expected or estimated to be at a level that's far below the sensitivity of our instrument. There will be need for us to concentrate organic compounds that come from the ocean world IC sample or liquid sample. But the question is, how can we potentially concentrate organic molecules without destruction, without bias towards a specific type of molecule? And most importantly, how can we design this instrument so that it can be universally compatible to any downstream instrument that we are looking at? So one way to do this is via bubble. The idea behind it is that when bubbles are generated from at the bottom via either fritz or from air bubbles, when they're passing through the column of solute, they will interact with whatever analytes that's currently in the bulk solution. So those analytes will be potentially attached to the surface of the bubble. And once the bubble rising up to the surface, the bubble collapses. All the material that's been stick around the outside of the bubble will collapse and eventually form a jet droplet with a highly concentrated material that's being dissolved in the solution. What we will do is basically to generate bubble, let it go through the bulk analyte solution, and then on the top, we will try to collect those highly concentrated aerosol or jet droplet. And eventually, by eluding those jet droplets to the downstream instrument. That's the primary ideas. This method actually has been demonstrated before, and then people realized the benefits of doing bubble physics is not only just to concentrate organics, but also can expelling those extra salt that's been present in the ocean world sample. So the advantage are squared. Currently, this prototype is still under development. And if you are interested in, I would be happy to tour around our lab and show you more progress. Okay, and that's the end of my presentation.